Hello.
Hmm.
So yeah, I was explaining this. Okay, so when a plant cell is immersed in a solution of lower water potential, that is a very concentrated solution, relative to its cell sap, water will diffuse out of the cell into the solution by osmosis, right? So I was talking about the consequences of this situation. Um, yeah. So what will happen? Obviously, the vacuole will shrink, right? So vacuole shrinks and the cell will stop exerting pressure on the cell wall, right? So the cell will become now even if after this happens, if you put it in a solution with a high water potential, turgidity of the cell can be restored. But then if more water leaves the cell, so if leaves the cell, the vacuole and the cytoplasm will shrink to such an extent that they will pull away from the that to such an extent that the cell membrane will pull away from the cell wall. I'm just going to show you what I mean. So in a diagram, if you look at it, you'll be easier to understand. Now, this is known as plasmolysis, okay? That the cell membrane pulls away from the cell wall. This thing is known as plasmolysis, okay? And this can be very lethal if the cell is not quickly transferred to a solution with a higher water potential, okay? So, yeah, I'm just going to show you a picture once you're done writing. Hello, are you done?
Yeah.
Okay, great. Mm, that's the problem. I don't know how to bring my picture down. Okay, here you can clearly see the different states of a plant cell. So we have flaccid, uh, mm, okay, we have flaccid and then turgid and plasmolyzed. So that is the one that is very critical. Here you can see even if some water is going out of the cell and the cell membrane is slightly detached from the cell wall, it is still there. But this one is completely detached from the cell wall, okay? And yeah, on this side we have turgid, which you already can understand. So I don't think you need to know these terms like hypertonic, isotonic, and hypotonic, but these simply mean they mean the water potential of the solution that is surrounding the cell. Okay.
Apu,
All
I
right.
had a question.
Yes.
Apu, when the cell becomes flaccid at first, then it is possible to get it back to the turgid state, right? But when it
Yes.
becomes plasmolized, it can't go back. The
Yeah.
cytoplasm and the vacuole has permanently changed shape.
Yeah. Exactly. It is permanently damaged, and like, yeah, when it is plasmolized, it will die like very soon.
Oh,
So yeah, it is a very critical stage.
yeah.
Okay. All right. Now we are going to move on to osmosis in animal cells. Yeah, so osmosis in animal cells. So basically, in the same way as a plant cell, if an animal cell is immersed in a solution with a higher water potential relative to its cytoplasm, then obviously water will diffuse into the cell by osmosis, right? So when relative to its cytoplasm, water diffuses into the cell by osmosis. Now, animal cells do not have a cell wall, of course. So the cell, what happens, sorry, the consequences, I'm going to write in red. The cell swells, of course, and if more water keeps entering the cell, it will swell to an extent when it will burst, okay? Well, that because it doesn't have a cell wall, there's nothing to protect it, right? And this situation, this thing is called cytolysis. I don't know if you need to know this particular term, but I'm still teaching you because I also taught you plasmolysis. So this is very similar, cytolysis. Okay. So let's say we have this animal cell which actually burst, and then all the liquid will come out of it. So that's a cytolysed animal cell. Okay. Um, yeah, what happens when we immerse an animal cell in a solution with lower water potential? In a solution with lower water potential, relative to its cytoplasm, of course, but I'm not writing it down. I hope you understand. Um, water will diffuses out of the cell by osmosis. And then what are the consequences of this? The cell shrinks, of course, and becomes dehydrated. Well, in particularly in red blood cells, little spikes appear on the cell surface membrane if they are dehydrated. And this cell is said to have undergone crenation. Okay. So in internet Okay, so the animal cell basically dies if it is not removed from the solution. It's almost the same as a plant solution, plant cell being plasmolysed. It's almost the same. Okay, so let's say if you want to see a crenated red blood cell, it looks like this. Okay, and then obviously inside we have this. All right, that's it. That's it with osmosis. Note them down.
Yeah. Up and down.
Okay, great. Um, okay. I think now we are only left with active transport, right?
Yeah.
What is active transport? Do you know?
I think it's the movement of ions, but it's from a low concentration to a high concentration.
Sorry again.
It's the movement of ions, right? But it's from a low concentration gradient to a higher concentration gradient.
Exactly, from a lower concentration gradient to a higher. It's like the opposite of diffusion, and that's why it requires energy, right? Just a moment, I'm trying to find a good video to show you active transport in. Mm, yeah, I think I found one. All right, let's begin. So if I want to write the definition quite formally, I think you have to write it like this. It is the process in which energy is used to transport substances across a biological membrane against a concentration gradient. So yeah, you're right, from a lower concentration to a higher concentration. It's important to mention that energy is used in this process, okay? Um, what else? How do we get this energy? Obviously from respiration. Now, can you give me an example of active transport, or is it too difficult?
I think it's the uptake of. Is it water from the roots?
Yes, very good. That's a very good example of active transport in plants. So that's how, waters and different mineral salts are absorbed by the root hair cells from the soil. So that is number one. Uptake of dissolved mineral salts, for example, by root hair cells. Another one in humans maybe would be the uptake of glucose by cells in villi. This is like, you'll see these are cells in our small intestine. So these are small like protrusions of the small intestine. Okay. So these are, the most common examples of active transport. Now, I want to show you videos of these two examples so that you actually remember them, and also it's easy to visualize how it happens, okay? Okay. Hello? Is it still working?
No, I don't think so.
I think it's time to join the other link, right? Hello?
So then
Okay, let's
then
just.
shall I leave from this one and join the other?
Sorry, what?
Then shall I leave from this meeting and join from the other?
Yeah, okay.